Hello everyone. So today's class is about pathogens. So this is the VCA study design. So the key knowledge, so the things that you need to know for this topic. So you need to know the role of the human immune system in detecting and responding to antigens. So the nature of antigens and their sources, self and non-self, and cellular pathogens and non-cellular agents, so like viruses and prions. So our learning goal at the end of this class is going to be you will be able to list various types of pathogens and explain how they cause disease. You'll know you'd be successful if you can list various types of pathogens and describe how they cause diseases, and you can differentiate between cellular and non-cellular pathogens. What are pathogens? Pathogens are cellular, example bacteria, or non-cellular organisms, example viruses, that cause diseases. Parasites. Parasites are a type of pathogen that complete their life cycle in an organism called a host. They are considered pathogenic if they cause disease in the host. So not all parasites are pathogenic. It depends on whether they actually cause disease or not. So examples of parasites are viruses and worms. Some parasites infect two different hosts in sequence: the primary host that is used for the adult stage and the intermediate host used for the larval stage. Some parasites are transmitted to their host via another organism called a vector. So the vector is basically what transmits the parasite. Endoparasites live inside an organism, whereas ectoparasites attach to the outside of a host. Okay, here is a little diagram. So what you need to do is try to identify the vector. This is showing the life cycle of a malaria pathogen. So what you've got here is a mosquito which bites a person, and inside the mosquito there's the malaria. Okay, so the mosquito, when it bites a person, sporozoites are injected in the blood that travel to the liver, okay, and those sporozoites then form merozoites, which go into the red blood cell, okay, and and form new sporozoites. And if another mosquito bite this person, that new mosquito would then now become infected as well. So what do you think the vector is? Is it the person or is it the mosquito? It's the mosquito. The mosquito is what's carrying the disease, the pathogen, and transferring it to the human being, okay? The human being, the person, will become sick from the pathogen, whereas the mosquito is not going to get sick from the pathogen. So the mosquito is only acting as a vector. Okay, so these are examples of ectoparasites and endoparasites. So the difference is one's inside, one's on the outside. You can see here, it's on the outside. So it's an ectoparasite. Disease. A disease is defined as any condition that impairs the normal function of an organism. So there's non-infectious disease. Those are the ones that are not transmitted horizontally from one organism to the other. Usually non-infectious diseases are hereditary passed vertically from parents to offspring. So they can be congenital, innate, or idiopathic. Idiopathic means people don't really know what the cause is. Then we've got our infectious disease. Those are the diseases that are transmitted horizontally from one organism to the other. So I'd just like to mention that vertical transmission is when the disease is transmitted from the parent to the offspring. And usually it's hereditary. Whereas horizontal transmission is from one person to another. Contagious disease. This is what form of infection disease transmitted by contact. Now let's move on to the different types of pathogens. Firstly, we've got our bacteria, the most common pathogen. Bacteria are cellular pathogen because they are cells, they are actual cells. They are prokaryotic cells. So their genetic material is not bounded inside a membrane or a nucleus. Bacterial cells have plasmid DNA or circular DNA. Some are motile. They have flagella, so this thing there, that structure there is a flagella, and the flagella allows them to move, while some don't. Some have a thick cell wall, okay? Now, bacteria that have a thick cell wall are also gram-positive bacteria, which mean they stain blue when you perform a gram staining. They look blue. And some have a complex multilayered cell wall. So those ones are gram-negative. They stain pink. It's the same stain, but they look different color based on what type of cell wall they've got. The cell wall is made up of a peptidoglycan, which is a biomolecule only produced by bacteria. Okay, so that cell wall you see there, it's made up of a special molecule called peptidoglycan. Okay, the cell wall is made of peptidoglycan, which is different to what the cell wall of plants are made of. Plant cell walls made of cellulose, whereas bacterial cell walls made of peptidoglycan. Some bacteria have endospores that allow them to survive harsher environments. Now they can sense and respond by developing a capsule and becoming dormant. So they can sense when the environments become harsher, and the way that they survive is by producing a capsule. And they become dormant, which means all their metabolic activities stop. Some bacteria require oxygen. They are aerobes. They're called aerobes. Some can live with or without oxygen, and those are called facultative anaerobes. And some cannot survive with oxygen at all, and they are called obligate anaerobes. So now what you need to do is pause this video so that you can copy down those notes. Welcome back. Now, bacteria, there are different shapes of bacteria. So you've got bacillus, which are rods, coccus, which are little spheres, and spirillus, which are spirals. You also get bacillococcus that are halfway between a bacillus and a coccus. So they're sort of spherical rods, really. They're in between the two, and they're called coccobacillus. It's not shown here in this diagram. Now, different species of bacteria arrange themselves differently. Okay, so here, when they are found in pairs, this is a diplococci. When they are just singular, it's a micrococci. When they form chains, they are streptococci. And when they form clusters, they are staphylococci. You see the same thing here with bacillus. So microbacillus, streptobacillus, And here are some bacteria with that one, for example, have endospores. So the endospores are basic spores that are on the inside of the cell. That's a bacteria with flagella. Those are spiral type bacteria with flagella on the ends, and that one on the one end. So there's different types, okay? There's different types of bacteria. Of course, not all bacteria are pathogenic, okay? Okay. The next microorganism we're going to look at are protozoan. So your next heading is protozoan. Those are also cellular pathogens, okay, but not all of them are pathogens. Some are pathogens while others are parasitical pathogens. Sorry, I'm making a coffee. Now, protozoans are eukaryotes, meaning they have membrane-bound organelles, unlike a bacteria, which are prokaryotes. So one type of protozoan that is pathogenic is sporozoans. So what they do is they can detect movement and they are attracted to movement. That's basically what causes malaria. And they are transmitted by mosquitoes, which is the vector. And that's what they look like there. And they detect movement. That's probably how they enter the mosquito in the first place, because they can sense movement and they get attracted to the movement, and then they just infect the mosquito. Then we have sarcodinians there, which engulf red blood cells and cause amoebic dysentery. So they infect red blood cells and cause blood-borne diseases. Okay, then you have something called Giardia, which causes diarrhea. And those protozoans have flagella, which means they can actually move. They also have like an adhesive disc that allow them to attach to intestinal villi, and that's what basically causes the diarrhea. Protista. Protista are cellular pathogens, which mean, again, they are cells. They are fungi-like in the sense that they produce spores and organize themselves similar to fungi do. However, they have a cellular cell wall instead of a chitin cell wall. So fungi have chitin cell wall. But those protista there, even though they look very similar to fungi, they don't have a chitin cell wall, they have a cellular cell wall. Furthermore, they are motile, they have a flagella, so they exhibit many cellular processes that are not found in fungi. So one type is the oomycete, which causes mildew on leaves. So if you ever see that, this is probably caused by a protista. So they like to infect plants, really. So they are pathogenic to plants. Then we have our fungi, which are also cellular pathogens. Fungi form spores. They have a chitin cell wall. They have antibacterial properties, which mean that sometimes where fungi grow, you won't get any bacterial growth there because they have antibacterial properties. In fact, antibiotics are derived from fungi. So only some types of fungi are pathogenic to animals. For example, Aspergilloma is a fungus that colonizes in a healed lung scar or abscesses from previous diseases. So the Aspergilloma is pathogenic, and it basically colonizes the lungs, okay? Then we have worms. So our worms are multicellular. So it's not just one cell, it's a lot of different specialized cells. You've got two different worms, platyhelminths. So those are known as flatworms. They are endoparasites, which mean that they complete their life cycle inside the host, and they basically live in the gut. They have a head that attached to the walls of the gut. They have a neck that grows and chain of segments that consist of the male and female reproductive organs. So basically, it's one worm that's both female and male. So those organs can fertilize each other, and once fertilization occurs between the segments, a bag of eggs is formed, which passes in feces. Okay. Then we have the nematodes. Nematodes are known as roundworms, hookworms, threadworms, or pinworms. They are present in huge numbers in the environment. They reproduce by sexual reproduction, but the offspring do not leave the host and cause severe disease in the host. So this is different to platyhelminth, where in the platyhelminth, the eggs leave the host and they don't really cause that much issue. But here, those one, the host, the offspring do not actually leave the cell and they cause severe disease in the host. They even have a complete digestive system and they can survive inside the host. Then we have our arthropods, which are also multicellular. So usually arthropods are transmitted by insects, vectors, such as mosquitoes and fleas. Okay, like if you've heard about fleas. So example of arthropods are ticks, mites, head lice, body lice, and crab lice. Now, talking about viruses. So viruses are non-cellular. Viruses are not alive, they're not considered alive because they are not cells. Okay, they cannot even reproduce outside of their host. They rely on the host to actually multiply. So they are non-cellular agents that can infect any cell type. They are specific though and can only infect certain types of cells. They are obligate intracellular parasites that cannot replicate without a host cell. This is because they do not possess the machinery required to reproduce, like they don't have any ribosomes to produce protein, they don't have any organelles, and what they have is their genetic material, which is protected by a protein capsid. Viruses there are some type of viruses that infect bacteria, and those are called bacteriophage. Now, bacteriophage is really useful in genetic technology, and we'll talk about that later on in Unit 4. There are two common types of viruses. You've got your naked virus that only have their nucleic acid, so their genetic material, protected by a protein capsid. And you have an envelope virus. They have their nucleic acid protected by the protein capsid, but they are found in a phospholipid envelope. Okay? So with those viruses, the genetic material can be either double-stranded DNA, it can be RNA, it can be single-stranded DNA, or double-stranded RNA. So here are the different structures. So you have a DNA inside the head, okay, and that head and the tail, basically, that's what the protein, that's made of protein. Okay, and you have that structure here, tail fibers, that's what the virus used to attach to the host cell. And then the DNA comes down there to infect, to go inside the cell. Or you have this type of structure here with the DNA on the inside covered by the capsid made of protein. The life cycle of a virus. Viruses can only infect specific cells that have a receptor molecule that match the viral attachment protein. So the virus have got a viral attachment protein. So usually those things there, those pins or those projections there, those are the viral attachment proteins. Now for them to be able to infect a cell, the cell need to have a receptor molecule that can attach to those viral attachment proteins. Okay, just like in signal transduction or just like enzymes work, they have to fit into the receptor. Some viruses do not have attachment proteins and infect cells via all the other mechanisms, such as being engulfed by macrophage. So macrophage is a type of white blood cell that engulf or eat pathogens. Okay, so when those macrophage go and engulf a virus, the virus can then enter that macrophage and the virus basically infect the macrophage, which is not a good thing. The virus life cycle. So once attached to the cell host, so here you see a virus that's attached to the cell surface, the genetic material is released into the cell. So you can see that here, the genetic material comes down and enters the cell. The capsid and envelope remain outside of the cell. Okay, so that bits there remain outside, but the genetic material enters the cell. The genetic material then enters the nucleus and merges with the cell's genome. So basically that material, the genetic material from the virus, combines with the DNA from the host cell. Then that takes over the cell's metabolic machinery to reproduce and produce viral protein. Okay, so here you've got your virus DNA that's replicated. It's used the cells like ribosomes and enzymes to produce multiple copies of the genetic material of a virus. Okay, so it uses the ribosomes to produce new virus components as well, like the head and the capsid and stuff like that. And those are called virions. So those here you see the virions. New form virions, singular for virus. Those then bud off the host cell and form a phospholipid envelope containing the host cell's recognition molecules, making it hard for our immune system to pick up on the non-self cells. So what happened here is they burst out, and in the process, they can cover themselves with a host cell's own membrane, and that makes it hard for our immune system to detect those cells. So this is basically what's going on here. Okay, so an influenza virus comes and infects the cells, produce several copies of itself, then it reorganizes itself to make the viral component, and then they leave the cells by budding off, okay? And it takes with it the cell membrane. Now, that makes it really hard for our immune system to detect the virus because they covered themselves with the cell membrane with our own recognition molecules on it. There's two types of viruses. There are lytic viruses and there are lysogenic viruses. Lytic viruses, basically, at the end of their life cycle, they just burst and rupture the cell open, which in the process, it kills the cells. So when they lyse the cell, they don't usually steal the recognition molecules. They just burst the cell open and they just escape, and they go on and infect neighboring cells. Those are lytic cells, okay? They lyse the cell. Over here are the lysogenic cells. Lysogenic cells are the one that they do not lyse the cell, but they form buds and steal the recognition molecule of the cell membrane. So there are DNA and RNA viruses. In DNA viruses, the DNA becomes part of a genome, whereas in RNA viruses, the RNA is used to produce proteins at the ribosomes. The RNA doesn't need to become part of a genome. Then you have retroviruses, where the RNA is transformed to DNA using an enzyme unique to the viruses called reverse transcriptase. Then the DNA becomes part of a genome. Viroids. Viroids are non-cellular. They are tiny circular single-stranded RNA molecules, about 1/10 of the size of the smallest virus. They do not have any capsid or envelope. They're basically free floating RNA molecules. They usually infect susceptible cells. They are called opportunistic pathogens. Opportunistic pathogens are basically pathogens that won't make you sick unless you're already susceptible. So far, they have only been identified to cause severe disease in plants. Then you have prions. Prions consist of proteins, prion protein PRP, without any nucleic acid. Some prions are pathogenic, while some aren't. Some prion protein PRP are normal, and some are disease-causing, called as scrappy PRP. Okay, so there's two types. There's the normal, and there's the scrappy PRP. Prions only infect cells that can replicate and code for new proteins. So basically, any like animal cells can do that. So the prions can infect animal cells. Okay. So the amino acids of normal and disease-causing PRP are similar, but the difference is in the shape. When normal PRP come into contact with disease-causing PRP, the shape of a normal PRP changes and it becomes a disease-causing PRP. Okay. In infected animals, scrappy PRP accumulates into the nervous system and slowly deteriorates the nervous system. Kuru in humans, mad cow disease, and scrappy in sheep are examples of diseases caused by prions. Okay, so next we will be talking about defending against pathogens, but that will be it for today. And I hope that this video has been useful. I'm now going to stop the video and I'll see you later.